Yeah, I think many of you will probably recognize Michelle. I know in the audience today visiting from Kansas Analytical, there's Matt Nethercott, and you see the PhD students who I've had over the years have been instrumental in making the research contributions and discoveries in the group, and going to tell you about some of those discoveries, including some made by Michelle and by Matt. For most of the time that I've been at Michigan State, one of the large projects in the lab has been understanding the joining, or the terminology that's used is fusion, of the membranes of envelope viruses with those of the target cells. So envelope viruses are a large class of viruses, including many viruses that cause important human diseases. For a long time, the kind of highest profile one was HIV, influenza. Now, recently, again, much more topical, is an envelope virus. Coronaviruses are Zika, measles, some cancer viruses. And sort of what they have in common is a membrane that they pick up from a host cell when they leave that infected host cell. And in order to infect a new cell, a required step is this joining process of the virus and target cell membrane. The important result at the end is that the viral genetic material, here it's represented by these dark triangles, which is typically RNA, is then inside the cell, and then there's the possibility of viral replication. So membrane fusion is a process where the free energy change is about zero, because you start with membranes, you end up with membranes. Really, it's just a physical reorganization or movement of the lipid molecules of the membrane. So there's no chemical or physical change of the membranes, but typically, as we know, as multicellular organisms, the membranes of our cells are not typically joining, and that's because there are large kinetic barriers to go from two membranes to a single membrane. So the viruses have peers independently developed a protein, or evolved a protein, to catalyze the fusion process. And it's actually part of the sort of larger spikes that are now iconic because of the COVID virus, but sort of all of these envelope viruses have these spikes protruding from their membranes, and part of the spike is a fusion subunit that catalyzes the fusion process. So I would say our major contributions to the field have been to elicit and determine the structures of much of the fusion subunit section of the spike, particularly the sections that bind membranes and that are most important in fusion catalysis. And then we've also been very focused mechanistically to understand how is it that this fusion subunit reduces the barriers that make the fusion and therefore the infection processes possible. So this is a schematic model of fusion, very schematic. I hesitate even to call it a model. It's really pictures, and there is some experimental evidence for some parts of the pictures. So the spike is has the fusion subunit that's colored here, and it actually has a transmembrane segment that goes through the virus membrane. And then these, there are structures for most of them, this sort of uh, dashed brown oval, those represent the receptor binding subunit, which is typically non-covalently attached to the fusion subunit 4. So the function of the receptor binding subunit is to bind specific molecules on the surface of specific cells. So, for example, for HIV, it's actually 2 proteins, a CD4 and then a chemokine receptor that are specifically found in specific immune cells, particularly T helper and macrophage cells. So the receptor binding subunit really identifies the cells that the virus can infect and where productive infection can happen. After receptor binding, in a step that's still not clear, structurally what's happening, but somehow the receptor binding subunit moves away from the fusion subunit. And in general, there's a section of the fusion subunit, 25 or so amino acids, that binds the target membrane. So that's really how the physical attachment to that membrane happens. Again, just want to emphasize that the amino acid sequences of the fusion subunit of different virus families are not related to one another. But this is a sort of general model then for how at least what we know about some aspects of fusion that seem general, though, to all these fusion subunits. Another sort of universal aspect of the fusion subunit is that without the receptor binding subunit, the part of the fusion subunit that's between the virus and the cell membranes changes its structure dramatically into a hairpin. And you can see, what you have with the one end, the C-terminal end of the fusion subunit in the virus membrane, the fusion peptide in the target membrane, and this is a very thermostable hairpin. You actually can bring the membranes close together, and I'll show you just a bit sort of why that's important. One other sort of feature that's this part is specific to HIV and also influenza. It's also common for coronaviruses, is that in fact the spikes are 3 copies of the fusion subunit and 3 copies of the receptor binding subunit. And we also know that the fusion subunit on its own can form trimers. So that's like right here, this is one fusion subunit trimer. There's another kind of how they're attached. I'll talk about the evidence for having these 2 trimer subunits of the hairpins sort of at the end of fusion. And then there's one more piece of information. So the fusion subunit of HIV is called glycoprotein 41 kilodalton, shorthand is GP41. The 41 kilodalton is a monomer molecular weight for a single GP41. So in terms of understanding mechanism, the approach, just as kind of general in biochemistry, is to make single amino acid mutations and then look at the effects on function, and then there's some interesting effects, then look at how the structure of the structural change because of the mutation, and then try to infer mechanism from the mutation fusion activity data. So there's been extensive work for this in GP41, and I want to highlight a specific mutation. It's B2E, so it's in the fusion peptide. this is actually the N terminus of the protein, so right at the end. This was first, it's an engineered mutation. It was first made actually in viruses. So that's the data I'm showing you here. It was first discovered about 30 years ago. So one effect of this V2E mutation is that really essentially eliminates the viral fusion and infection. There's no more fusion and infection. It's also phenotypically dominant. I think, as far as I know, it's the only mutation where this has been demonstrated. And specifically in these tables, you're looking at viruses which have mixtures of wild type and V2E proteins. And you can see that if you have spikes that have 10 wild type for one V2E, you only get 40% of the fusion and infection. So the loss of fusion and infection from V2E is phenotypically dominant. So besides using the phenotypic dominance and additional studies to understand mechanism, it's also been important in trying to estimate the number of wild type spikes that are needed for efficient fusion and infection. So again, mechanistically, and also that's interesting in terms of developing an HIV vaccine, which we still don't have, but also therapies and therapeutics that can inhibit fusion. And I'm just showing you this table. They're all using the same data and just different models to estimate T, which is the number of wild type primers needed for efficient fusion and infection. And the differences in the values of T goes from 1 to 19. It's really just the differences in the statistical models to analyze the experimental data. So mechanistically, I was interested in knowing whether this phenotypic dominance would actually carry over to just GP41 induced membrane fusion, which our lab had studied for some time. It turns out, if you make GP41, it by itself, it forms very stable hairpin trimers. It will induce vesicle fusion, so it's good at membrane fusion. Always a question that I had that one should have is what we were studying the test tube, in fact, reflective of the virus, which, it's a virus, there's cells, if it's potentially quite different. So kind of PCR data now from our laboratory, and sort of the short answer is yes, and in fact, the phenotypic dominance matches quantitatively as exactly as one could reasonably expect. So what I'm showing you here now is vesicle fusion data. So, and then that's the vertical axis, time is the horizontal axis. At time 0, we add the protein. You see the black curve is for all wild type. And then the other curves, we add in some V2E. So you see this light green curve, we have 19 wild type for 1 V2E, and already the fusion is going down. This dark green is it's 9 wild type, 1 V2E, and we're actually very close to the red line, which is all V2E. So we see this phenotypic dominance. And the other effect that I was not expecting this effect at all. We can use an approach called circular dichroism spectroscopy. It's an ultraviolet spectroscopy, and in particular, the depth of the signal by conventions at 222 nanometers tells you how much helix you have in a PP41. Essentially, all of the helix in the GP41 is in this hairpin region. So you really, this measurements are telling you about the length of this helical hairpin. And the black data, again, is all wild type. Red is V2E. And you see about a 20% loss in helicity. And the V2E is also phenotypically dominant. So the green here, so that's 9 wild type for 1 V2E, and you're already about half of the way, or half of the loss of the hairpin helicity. This was really surprising to me because the fusion peptide segment we knew from work in our lab, and the hairpin segment from work in other labs, so their structures form independent of one another. So it's completely not clear why would a fusion peptide mutation affect the structure of the hairpin. It's clearly some kind of allosteric effect. We actually plotted all of the viral and R data. Here I won't talk about the analysis in detail, but we were able to analyze all the data together to show that you need 6 wild-type GP41 to get efficient fusion and infection, and that's 6 wild-type GP41 is 2 trimers, and now you see some of the experimental data from our and other labs that supports that picture I showed in a previous slide of the 2 trimers. So kind of in the next part of the talk, I wanted to address really what is the structure, or what are the structures of this fusion peptide segment in membrane. It turned out to be, I guess, both more challenging and much more interesting than I imagined going in. And also kind of this fundamental question, why would a mutation that's 25 amino acids away from the hairpin actually change the hairpin structure at all? And clearly those 2 things together should help us to develop a real molecular-based mechanistic model for how the GP41 catalyzes fusion. So I wanted to now move into the NMR section of the talk. So this is just a schematic of the sample preparation. So we either chemically synthesize fusion peptides, or we use molecular biology to make the larger GP41 protein. We add it to aqueous solution with vesicles. In many cases, we actually can see the fusion in the tube because the vesicles get bigger, they scatter light, so they get cloudier. And then we do centrifugation, and the NMR is done on the pellet after centrifugation that has the lipid and whatever peptide and protein attached to it. Just point out, this is an all aqueous sample preparation. With membrane proteins and NMR, sometimes one uses organic solvents. We were trying, as best we could, to recapitulate how the virus is fusing with the cell, which of course is an aqueous medium without any organic solvent. So actually, it's been a long-term project in the lab. These data are actually from Michelle, when she was a PhD student working together with me. So the first question we wanted to address was the secondary structure of the fusion peptide in membrane. Basically, is it mostly a helix or wound up? Is it extended as a strand? The way we did this was to use the dependence of the carbon chemical shift on either being helix or strand, and these tables plot the differences between the experimental shifts, carbon shifts, as you go across the amino acid sequence, and the shifts expected for either helix or strand. And you can see we get much better agreement with the strand chemical shift. So we have these extended structures using the chemical shifts and other NMR constraints from our lab. We have the sort of medium resolution structure of the fusion peptide in membrane. And then we did additional experiments. So typically always if you have an extended beta strand structure and we knew we were the peptide is in the membrane with you most likely form, and we did NMR experiments to demonstrate is a beta sheet. And in fact, our beta sheet is an intermolecular beta sheet. So it's held together through these backbone hydrogen bonds. So I'm just showing schematically fusion peptide 1, 2, 3, forming this intermolecular beta sheet. So when you have any beta sheets, so you have multiple strands, and the next structural question is how do the strands that are hydrogen bonded to one another, neighboring, how do they line up? And so that in general is called the registry. So to general classifications are parallel, where the strands line up kind of in the same direction, or the different molecules line up in the same direction. You could also have anti-parallel, as I'm drawing here. Say me and a couple different graduate students really focused at first on this in-register parallel beta sheet structure, and that's because there were many NMR data on amyloid fibrils, which are also intermolecular beta sheets. And all in amyloid fibrils, these are what's deposited in the brains of people with neurodegenerative diseases like Alzheimer's, Parkinson's, Huntington's, etc. So those amyloid intermolecular beta sheets, they're always in register parallel. Many experiments, we don't actually have any in register parallel. We have completely anti-parallel beta sheets. So if you have anti-parallel, then there's another potential variation, which is called registry. So this is really sort of which amino acids are hydrogen bonded on between the adjacent molecules. So I created an index, I'm going to call it little t, and it's simply the length of the beta sheet. So here you're seeing a t equals 17 anti-parallel sheet. This is t equal 20. So kind of the next set of experiments was to see if we had a single value of t, or do we have a couple of values of t? Do we have, it turned out we have, our sheets actually have many values of t. So there are actually many different anti-parallel beta sheet structures. To get at this, originally, we would say for the conventional NMR methods where you detect intermolecular or internuclear cross peaks and look for proximity between nuclei, which would be consistent with a particular registry. And we definitely saw cross peaks, but it's clear after collecting more data that a single, we couldn't understand our data with a single or even a couple registries. So really the successful approach is one called rotational echo double resonance, or REDOR. And our samples, we have just a single backbone C13 carbonyl label, like one residue and a single nitrogen 15 label at a different residue, these carbon-14 and nitrogen 15 are far enough apart where we don't see any intramolecular effect. What we could potentially see for the population of a registry that lines up the C13 and N15 is what's shown here, this reduction in signal and of the carbonyl signal and what will call the S1 relative to S0. This is a dipolar coupling effect. You see it when the CN distances are less than 8 angstroms. So if you have this alignment, as I'm showing here, if you're hydrogen bonded, you've got a 4 angstrom distance. These carbon and nitrogen are not hydrogen bonded, so that's 5.5 angstroms. But you will definitely see this loss in signal. And the data are plotted as what I'm calling a dephasing. So that's this delta S reduction in signal divided by the full S0 signal. So that typically will vary between 0 and 1. We also vary a parameter, which we call the dephasing, or you call it the evolution time. And typically, if your carbon and nitrogen are less than 8 angstroms apart, the dephasing will increase with dephasing time. Very importantly for our work, your dephasing at long dephasing times comes to some asymptotic value, which is approximately the fraction of molecules in your sample where the carbon and nitrogen are less than 8 angstroms apart. So this, and then should also point out that another great advantage of REDOR is that if you know the geometry of your spins, which we know this for a data sheet, you know it for an alpha helix, you know the geometry of your spins for your labeling in a particular registry, you can quantitatively calculate dephasing as a function of dephasing time. So this is for a model compound. The little X's are our calculation. The brown circles are the experimental data. So that's the ability to use quantum mechanics to simulate the dephasing is a very important feature of REDOR that we use. Yeah, so this simply summarizes the REDOR experiment. And now what I have to say, describe the data that we collected. So as I said, we have a single backbone carbonyl. So here it's at leucine 12, N15 label, it's an alanine 6. And then I create now a labeling parameter for that particular labeling pattern, which I call U, which is the value of T that lines up your carbon and nitrogen labeled residues. So when, or for the fraction of molecules where T equal U, right, you will line up your carbon and nitrogens, and you would expect substantial dephasing. Just point out this is now the U equal 20 sample, so I have moved the positions of the label. And we make many different labeled samples with different values of U to determine these registry populations. So one complicating part of the analysis is that there are not just the T equal U registry that contributes to dephasing, but actually T equal U plus or minus 1, and also plus or minus 2. So, for example, I'm showing schematically the U equal 17 sample with the T equal 18 registry. The distances are longer than U equal 17, T equal 17, but still less than 8 angstroms. So you can't directly relate the data for a single sample to a single registry, and that ends up being important in the analysis. So it's a really kind of heroic effort by two PhD students, Scott Schmidt and Li Shi. They made actually 18 differently labeled wild type and 18 different labeled B2E samples and did the read or NMR. You'll see the data in just a little bit. And just kind of remind that the goal was then to take these data and determine registry populations for the wild type beta sheets and the B2E. So I know this is a lot of data. I just wanted to give you a sense of the data that's being analyzed. This is the data from 40 millisecond dephasing time. So we're close to the asymptotic limit. We've got S naught in blue, S1 in red, and then wild type in B2E, and then we've got U equal 8, 9, 10, 11. So all the data for 40 milliseconds. So again, besides making all the samples, another heroic and really important part of their work was to obtain really high signal to noise data and see how good it is. And I'll just sort of guide you through qualitatively what we learned just from some of these data. So we'll start with the U equal 8. You can see there's very little dephasing for either wild type or B2E. So just qualitatively, that means we don't have very much population of registries near T equal 8. So for U equal 13, now you can see, right, there's a large loss in signal for wild type, much less for B2E. So that means for wild type, we probably have some significant population of registries of beta sheets sort of near T equal 13, less for B2E. For U equals 17, it becomes, you see, similar dephasing for wild type and B2E. So they, both wild type and B2E, have some population of registries near T equals 17, and then 19, it reverses. Now you see B2E has much larger dephasing than wild type. So, teams, just looking at some of the data, that there will we definitely have larger populations of longer beta sheets for B2E. I get more generally, stare at the data for a little bit, you'll see that kind of many samples are showing dephasing. It's never close to 100%. So what that's, again, qualitatively telling us is that we have many populated registries for wild type and B2E, but the dephasing is different. So they both have these sort of broad distributions of registries, but there's this weighting towards longer beta sheets for B2E. So now here's more data, but this is actually the data that we analyze. So again, I'm actually showing the dephasing as a function of dephasing time for wild type and B2E. Again, U equal 8, 9, 10, 11, 12, 13. One thing I've added is these dashed lines. So there's a natural abundance. Carbon 13 is 1%. Nitrogen 15 is 0.4%. So you're going to always get some dephasing because some natural abundance carbon 13s will always be near natural abundance nitrogen 15. And we can actually calculate that from the structure and the spin geometry. And that's what's shown here in these dashed lines that are the same on all the plots. And you can see for U equal 8, the dephasing that we do see for either wild type or B2E is all natural abundance dephasing. For U equal 13, so these points here, so that's the wild type. And you can see that there's much more dephasing for U equals 13 than expected from natural abundance dephasing. So we can really qualitatively see that we have some significant population of registries near T equal 13 for wild type, much less so for B2E, which is down here, just kind of a long dephasing time is just starting to get above natural abundance. For the U equals 17, you can see both the B2E here in green and the wild type in magenta. So they're both well above the natural abundance dotted line. So there are a significant population of T equals 17 or close to T equals 17 registries for both wild type and B2E. And as expected from our observation of the spectra, so for U equal 19, you see now the wild type, sorry, the B2E has much larger dephasing relative to natural abundance than wild type. So the wild type, is still clearly above the what's expected from natural abundance in the dotted line, but again, a more qualitative evidence for weighting of populations to longer beta sheets for V2E. We had these data for a long time. I kind of worked on the analysis on it. Then I was very fortunate to have a new PhD student, Yijin Zhang, who was interested in programming and fitting and she taught herself Python code. Always I knew kind of my goal, really the important goal of the data analysis was determine these fractional populations for different registries T. So I'm going to call this F of T. The complicating part is that for a particular sample with some value of U has contributions from the population of the T equal U registry, but also U plus or minus 1 and U plus or minus 2. But we would expect that all samples, independent of labeling, should all have the same fractional populations. So the only thing that had to be done, and this is where the programming was so important, was to fit all the data simultaneously to get a single set of FT populations. So just wanted to show, this is the master equation that I developed. So on the left, you see we got the fractional population. It's actually a sum, both from U equal minus 2 to U plus 2. What's here in brackets has particularly these gamma T's. We calculate these from a quantum mechanics-based spin physics program. This is important because for a particular value of U, the T equal U registry will have the biggest dephasing, and then U plus or minus 1 will have less dephasing, and plus minus 2, even smaller dephasing. So that's accounted for in these brackets. And then on the right side is the experimental side. This is our experimental dephasing, particular sample U at a particular dephasing time. And then this quantity here in brackets, that's the natural abundance dephasing that has to be always subtracted out in doing the data fitting. And it worked. High quality data, doing a careful analysis. So what I'm plotting here are our experimentally determined fractional populations for wild type in blue, B2E in red. So kind of all the qualitative conclusions from just looking at the data, how it pulled up. You can see you have, these are our registry numbers here along the horizontal axis. Many populated registries for wild type in B2E, definitely a weighting towards longer registries, longer beta sheets for B2E. Say this work actually just got published Monday, 2 days ago in PNAS. I'm very excited and proud about that. The fitting was excellent. Particular, the chi-squareds are close to the number of data. That's really the metric that your model is a good one and that there's no anomalies in your data. One really is quite surprising conclusion was that you see that the populations don't change continuously with T. So like wild type, you see T equal 13 and 15 are large and 14 is small. For either wild type or B2E, you see 18 and 20 are large, 19 is very small. So then I thought, well, okay, I guess the other important experimental set of experiments we did was to remake samples with, I guess the peptide had the same labeling, but we resynthesized the peptide, made a new sample, retook the NMR data, and within error, the data fits among replicate samples. And I think this really points to that our distribution of registries is a thermodynamic distribution. So it's the lowest free energy distribution. And that potentially there should be then some free energy function as a function of T that would then explain kind of all these different populations. And then, of course, if you have many populated structures, the different structures have to have approximately equal or similar free energy. So that is a necessary feature of the free energy function. So I tried many different free energy functions as a function of T, and the one that I'm showing here is the one that by far works the best for the wild type data. So there's 3 free energy contributions. So one is simply the length of the beta sheet, that's here in red. There's the fusion peptide is in membrane, so there's insertion of side chains, that's shown here in purple. And then to get kind of that up-down 13, 14, 15, I needed to incorporate, you could call it a leucine steric zipper. So that's, you get a negative free energy contribution when for the registries where the leucines on the adjacent hydrogen bonded peptides line up with one another. So this has been observed in other protein structures. So the leucines actually form a tightly bound or tightly packed structure, and so you get a negative free energy. And kind of why is the free energy similar for many different registries? So you see that the beta sheet length clearly, So the blue here are the fitted populations from the NMR data. The magenta are from the free energy function. And then the negative here, these are our different free energy contributions. So the beta sheet length, you can see that gets monotonically more negative as T gets larger. But importantly, the purple membrane insertion contribution is very weighted towards smaller values of T, so shorter beta sheets. And so the two contributions compensate for one another, so the sum is approximately equal or similar for many values of T. And then you can see the up-down really is a manifestation of either having, it's shown here in green, either in having the leucine steric zipper, like in 13 and 15, or not having the aligned leucines like in 14. And similarly, you see 18 and 20 have the aligned leucines in the steric zipper, and P419 does not. So the D2E was fitted by a similar function. The only thing that wasn't needed, or when I included in the fit, it always gave me zero contribution, is the membrane insertion. So for V2E, we only have beta sheet length and leucine alignment contributions. And this is completely consistent with other data from our lab that the V2E is much more on the surface of the membrane than wild type, which is actually quite deeply membrane inserted. And then some work that Matt Nethercott contributed to, we also did some analysis of much more full-length GP41 samples and membranes, and in particular for those full-length GP41 with fusion peptide. We did NMR experiments to probe the registry population in the full-length GP41, which is a sort of course, much more like the GP41 in HIV. And really, generally, there is very good correlation between the populations of the fusion peptide registries in the full-length GP41 as was observed with only the fusion peptide, where we did a more complete population analysis. So, to my knowledge, this is at least the only example I know whereby experiment a very broad, it seems like more than 10 different structures and their populations were quantitatively determined for a single sample. So usually in NMR, you can quantitate populations where you have resolution of chemical shifts between different structures. That is not at all the case for the fusion peptide. Yeah, I guess you can sometimes grow different crystal forms, but again, you typically only get a single structure from that. And my examination of the cryo-EM literature, I mean, sometimes you might get might get 2 populations of structures from a sample, or maybe 3. But to my knowledge, this is the first determination by experiment of populations of a very broad structural distribution. And it's, then got to think, like, why did the HIV virus evolve to do this? And I'm going to sort of tell you my hypothesis, model, theory, and kind of the evidence that supports it. The one thing that's, I guess virus infections in general get divided into chronic infection like HIV. So even without any medicine, a person infected with HIV can live for 5 years with the virus continuously replicating within that person. So kind of over those 5 years, the virus is continuing to infect new cells. And then there are any viruses like COVID, influenza, measles. So there, the person's infected maybe for a couple of weeks, and then the virus is completely cleared from the person. So in HIV in particular, with infection, there's a massive immune response, including against the fusion peptide. And kind of like all viruses, like the kind of the viruses, not like it thinks to do this, but the virus amino acid sequences are continuously mutating or changing during the viral replication. the immune response, particularly antibodies, are typically very specific to a particular amino acid sequence. So some of those mutated viruses aren't destroyed by the antibodies in the immune system, and those can replicate. So that's, HIV is continuously mutating as it's replicating, and kind of that's the way that it can be a chronic infection in a single person for 5 years. However, it also has this major challenge that if the fusion peptide has to remain functional. Fusion peptide is quite important in fusion. Many mutations, like I showed you for B2E, you don't get any fusion at end infection. So the virus has these sort of competing requirements of always mutating to evade the immune system, but also still being able to do fusion. And I think that a very broad distribution allows the virus to do it, or sort of the evolutionary solution, because kind of looking at these different linked beta sheet structures, I think plausibly many of them are at least good enough at doing fusion. So you can imagine that at least some of the mutations in the fusion peptide will simply shift the populations among these different functional registries, but also some of them then will also lead to viruses where the antibodies in the immune system no longer work. So I think the broad structural distribution is really the virus solution to these two competing requirements of immune evasion and fusion function. Okay, so what's the evidence for my hypothesis? Well, influenza virus also has a fusion peptide, also very important in influenza fusion. Amino acid sequence is not related at all to that in HIV. And influenza is an acute infection. So basically its strategy is just in that sort of couple days between one being infectious and one having symptoms of a person through aerosols infecting another person. So acute infection, not a chronic infection. And its fusion peptide structure determined by our group and other groups is really very, very different from the intermolecular antiparallel beta sheet of the HIV fusion peptide. So in particular, the fusion peptide formed these individual little helical hairpins, no beta sheet. There are two sort of very closely related hairpin structures, but certainly not 10 structures. Also strikingly, the influenza fusion peptide sequence is almost completely conserved. It's almost completely the same across all of the many subtypes in the flu world that they use for variants of the influenza virus. So like bird flu is H5N1, so the H is the hemagglutinin, which is the GP41 of the flu spike. H3N2, the H3 is that's the seasonal flu virus. So there's almost complete sequence conservation among the acute infection influenza fusion peptide sequence. You look at the HIV fusion peptide sequence among infected individuals, and particularly recently infected individuals, there's very broad amino acid sequence variation. And so I think this lack of sequence conservation is really related to to a continuous mutation that's happening within a single individual before, unfortunately, they infect another individual. Just sort of, I guess, want to really highlight again, I think, interested, I feel like, or my understanding of literature, this is, I guess, not, one of the first, if not the first, quantitative determination of a very broad distribution of molecular structures by experiment. It's kind of interesting, of course, structural distributions are commonly determined in molecular dynamics simulations, but it's unclear often whether that's actually the thermodynamic distribution. So that's kind of a more methodological contribution. I will just, I'm at sort of end of my time. Just put everything together. So what I want to convey on this slide is sort of how do these longer beta sheets for V2E, why do they lead to these shorter hairpins? And so I think that is due to the fact that there always has to be a connection between the end, which is the C-terminal end of the fusion peptide, and the N-terminal end of the helical hairpin. So if you make the beta sheet longer, the connector sequence stays the same length, and you necessarily have to shorten the helical hairpin. Okay, this is, I think, an important, really important reason, the shortening of the helical hairpin for why the V2E mutation leads to viruses that are terrible at fusion and infection. So if you make a shorter helical hairpin, sort of necessarily by geometry, you can't hold the virus and the host cell membranes as close together. So this, having the virus and host cell membranes further apart will really reduce the rate of membrane fusion, because at least by calculation, the largest barrier in fusion is simply bringing two membranes close together. And so if you hold them further apart, they will be much slower at fusion. So I think that sort of brings together the sort of earlier part of my talk and then the work on the structural distributions of wild type and V2E. So I think with that, I will stop. Thanks very much for listening, and I'd be happy to answer questions.
So in doing these experiments, you would kind of inherently limited by intramolecular transfer.
Yeah, if you do that, then your data will be completely dominated by intramolecular, which is, we have done some of those experiments, not here yet, if you want to learn about the intramolecular structure. Yes.
So should you do it by
Yes, and we. Yep. No, yes, we actually did those, that kind of labeling first. That was actually work done by Scott Schmick. And it was really through that approach that we showed very, very clearly and with the analysis that we only had anti-parallel beta sheet and we had none of the in-register parallel beta sheet. So we did do those experiments. The complicating part, though, is of course then you have to decide. I want to have this fraction of carbon-13 labeled peptide, this fraction of nitrogen-15 labeled peptide, and of course then you're in your data sheets you have to then take into account the statistical. You have to include the statistics. There's going to, like if you do 50-50, then there's a 50% chance that carbon-13 is next to nitrogen-15. There's a 25% chance it's carbon next to carbon, and so anyway, yeah. So we did do that. We did it in the way, putting the two labels on the same peptide, but far enough apart that they would not contribute to the readout signal. because then in the analysis we don't have to take the statistics that I described into account in the analysis. Yes.
And how much I think is important. When you compare the samples, you at least
Or they you know, we have the vesicles, then we add the protein, and then they bind. Centrifugation, yes.
And so is that, why can't you just it's hard to do the experiment if it's not taken.
Yeah, there's two reasons, NMR reasons. So one is low sensitivity. So in the way you're describing, that of course then your concentration of peptides or proteins is much more dilute. That's one reason. And then membrane vesicles are like basically typically you get very broad signals by NMR because you're in this terrible emotional regime. You're not in kind of the liquid state regime where you have a rapid enough tumbling to average out the anisotropic NMR interactions, but you're also not in the solid state regime where you're at least rigid or close to rigid. I mean, I guess you're fundamentally in this region of intermediate exchange where you get very, very broad lines. So sort of low sensitivity and broad and often uninterpretable signals is why we didn't do that. And very, I mean, so in the membrane protein world, you'll see there's, at least by NMR, there's very little work done in vesicle. So then what is the membrane mimetic? So for a long time, it was detergent micelles simply because it's small enough that you could still get the rapid tumbling. Some work on bicelles where you typically you still got lots of detergent there. In nanodisc, there's a little bit of work, but it's all kind of you get bigger in whatever sort of object that has the lipid, then the tumbling slows down and lines broaden and you're in this intermediate exchange.
So you can't make the vesicles smaller.
No, and then, well, I don't know if it's a real criticism, it's a study section criticism. Kind of if you go to small vesicles, then what you end up with is very, very highly curved, very, very highly curved vesicles, which at least it's an easy study section critique that your membrane curvature is completely not like and is having some unknown deleterious impact on your protein and your experimental system.
Your schematic looks like the peptides are they're like, what is the sort of relative surface density of the peptide?
Yeah. That's not true. So typically, I would say we would have something like 100 lipid molecules for every peptide. So it is absolutely true that that a single vesicle, which there are size vesicles, they probably have 100,000 lipid molecules. So they will certainly have probably multiple data sheets. I can say so the data sheets we don't have a direct or quantitative number on the number of strands. The data we do have, I would say, is maybe 20 data sheets. And it could be 6, but are 10-ish molecules for beta sheets. You definitely will have multiple beta sheets, separate beta sheets on a single resin.
And then down the solid state at the bottom, it's not likely that you have protein actions from. But are you measuring, especially for this ETP where it's more membrane critical, sort of measuring that in like this which state of where it's on the surface of one membrane and then push it up against the surface of another? Like I'm just trying to picture what the system looks like.
Sure. I mean, of course, probably in the centrifugation process, you don't have vesicles anymore. Right. I mean, probably honest answer is I don't know. All the data we have, I think what you're describing is unlikely. I mean, given kind of the peptide to lipid molar ratios and then the dimensions of the data sheet, dimensions of the lipid, I predict that we have fairly separated data sheets, but I don't have definitive data to support that. That's what I think. I'm definitely worried about like the beta sheet contact. I mean, I think I can say in beta sheets where structures have been determined, and it's that beta sheet contact. Yes, I think that the backbones of the two beta sheets would be about 10 angstroms apart, so that for our reader measurements will not contribute to the reader defacing. Because we're really, because we're focused on the interstrand hydrogen bonding. It's really it's 4, 5, 6 angstrom carbon nitrogen distances. So 10, and then I guess this is now a property of any dipolar coupling. It goes as 1 over the distance cubed. So 1 over 10 angstroms separation has 8 times smaller dipolar coupling versus 5 angstroms. The 1 over r cubed dependence of the dipolar coupling really helps to attenuate couplings due to more distant carbon-nitrogen pairs.
First of all, great seminar. So are you still using the 90-tumor, the 6-dose fundamental, and the 6 residue linker? What's like the work that you did for the wild type versus B2B?
Actually, it's longer now. So yeah, we include the whole membrane proximal external region. So in the work I showed here, we don't have the transmembrane. For this work, although we have prepared, and we not with B2E but with wild type, we have done experiments with GD41 that includes the transmembrane.
Quick follow-up. So then, are you still doing the neochemical ligation, or are you doing the whole thing in the?
No. I think Matt's referring to like we and Matt and some individuals in the group kind of the maybe it isn't so clear. It's kind of the now I can explain it. So the fusion peptide line widths, they were always much broader than would be typical for a protein with a single structure. And studies actually predicts, well, they sometimes say they would still fund me, but I always got commented on. Now I can say the reason lines are broad is because you typically get sharp lines with a single structure. We have 10 different structures, they have slightly different fundamental shifts. That's why the lines are broad. The problem, of course, any kind of spectroscopy, you have broad lines and you have multiple signals, and resolution of signals of distinct signals becomes much larger. So in NMR, we're looking, like in our case, at carbon-13 or sometimes nitrogen-15. But if you label all the carbon-13s, like in the whole TP41, you'll see a big signal, but can't distinguish typically which part of the signal comes from the fusion peptide or the hairpin or the transmembrane domain. So kind of what Matt and other individuals in our group did was to you can chemically synthesize the fusion peptide and put isotopic labels wherever you want. You can have minimal number of labels, and you can make by expression the rest of the protein, and then this native chemical ligation, the native part is in, you get a full protein, but it only has limited labels. Long answer, yes, we have published in that area, but it's I guess to do that, right? The thing is you have to do the peptide synthesis, which is fundamentally organic synthesis. You have to make the protein, which is fundamentally molecular biology, protein expression, and the native chemical ligation, which it's a kind of chemistry that certainly been worked on for 30 years. But as Matt well knows, like you have some published protocol, you do it and it yields 0. So it seems we start on our hands every single system, to put together the fusion peptide and the rest of the protein. You have to screen many conditions, and I guess I haven't had students like you who are willing to do that sort of sense. Anyway, long answer to you.
Thank you, David.
Yeah.